Hello, I'm Troy, and welcome to the Arcata Community Forest. The Arcata Community Forest and the adjacent Fern Lake provide a rich environment for studying microscopic life. They are characterized by a mixture of shaded woodland and open water, fostering a variety of microhabitats. I collected samples of moss and lichen from trees within the forest, which I then immersed in distilled water for several days to facilitate the release of microorganisms into suspension. I also took a separate water sample from Fern Lake. Standard laboratory techniques were applied to ensure that samples remained uncontaminated during transport and analysis. Overall, the Arcata Community Forest and Fern Lake offer a dynamic backdrop that reflects both terrestrial and aquatic microbial communities, making them an exciting choice for the scientific examination of microscopic life. I used my compound light microscope, equipped with a camera adapter attached, to shoot footage with my camera. I made a filter with a piece of plastic packaging and a dime for the dark field and oblique shots. The observed amoeba, a single-celled organism, exhibits a flexible shape and pseudopodial movement when feeding. Under the compound microscope, the amoeba displayed distinct cytoplasmic streaming and irregular outlines, common features among protists. A recent article titled, "This Amoeba Eats Prey Like Owls Do," describes a similar organism named Strigomyxa ruptor. That protist belongs to the vampirillid group and hunts in a manner that is unique among amoebae. The organism actively engulfs entire algal cells, enclosing them within a specialized chamber where digestive enzymes partially dissolve the prey's cell wall. As one side of the wall breaks down, the remaining portion stays attached, allowing the amoeba to extract the cellular contents before discarding the empty shell. The detailed observations confirm that such feeding behavior, though intricate, follows a pattern that appears adaptive for environments with limited food availability. Gastrotrichs, minute, worm-like animals, were observed in the prepared samples, commonly referred to as hairy bellies or hairy backs, relatable. These organisms are characterized by their elongated bodies and a covering of fine cilia that aid in locomotion across substrates. The specimen examined here shares similarities with the newly described species, Rhytidaceus brasiliensis, which has been recently documented in Brazilian inland waters. Detailed investigations of this gastrotrich species employed an integrative taxonomic approach, combining advanced microscopic techniques and molecular analyses. These methods confirmed that Rhytidaceus brasiliensis is distinct from previously known freshwater gastrotrichs. Phylogenetic trees constructed from these molecular data illustrate that although the new species exhibits several external morphological traits common to its genus, differences in ventral ciliation patterns and muscle organization set it apart from its relatives. Furthermore, the study highlights that the colonization of freshwater habitats by members of the Microdacida order appears to have occurred only once. This finding suggests a singular evolutionary event that enabled these organisms to adapt to a freshwater environment, diverging significantly from their predominantly marine counterparts. Lepocinclis, a genus of photosynthetic euglenids, was identified in the collected samples. These single-celled organisms possess plastids that are integral for photosynthesis and display a characteristic spindle shape. A recent genomic study titled Dynamic Evolution of Inverted Repeats in Euglenophyta Plastid Genomes focused on the dynamic evolution of plastid genomes within the Euglenophyta. In particular, it highlighted that species of Lepocinclis, along with related genera, exhibit notable variability in their plastid DNA. The study explored the presence of two inverted repeat regions carrying the rDNA operon, a feature absent in some other euglenid groups. Variations in gene content, arrangement, and the occurrence of maturase genes have been linked to differences in intron numbers within these genomes. Such findings indicate that the evolution of the plastid genome in Lepocinclis is an active process, driven by repeated rearrangements and gene loss. This dynamic evolution suggests that even among closely related species, there can be significant genetic diversity, reflecting adaptation to varying environmental conditions. Rotifers, small multicellular animals known for their resilience, were also abundant in the samples. These microscopic organisms are noted for their ciliated structures and a body plan that supports both feeding and locomotion. The rotifers observed resembled belloid forms, which have garnered attention due to their remarkable ability to survive extreme conditions. A notable study has reported that certain Arctic rotifers can remain viable after being frozen for as long as 24,000 years. In that research, samples extracted from Siberian permafrost were carefully thawed, and the rotifers demonstrated the capacity for parthenogenic reproduction once revived. Parthenogenesis is where an embryo develops directly from an unfertilized egg. Laboratory experiments revealed that these rotifers enter a state of cryptobiosis during prolonged freezing, where metabolic processes nearly cease. This state is maintained by specialized mechanisms that prevent the formation of damaging ice crystals. The study emphasizes that the rotifers' ability to survive extended periods in a frozen state not only highlight their unique biological adaptations, but also provide valuable insights into the processes of cryptopreservation. Roundworms, or nematodes, are parasitic organisms that have been linked to various health effects in larger animals. The microscopic specimens identified in the pond water included a species that exhibit characteristics similar to those of Ascaris. Detailed research has established a connection between exposure of parasitic roundworms and reduced lung function, particularly in younger male subjects. A study titled "Roundworm Linked to Lower Lung Function and Asthma in Younger Males" reported that individuals with evidence of Ascaris exposure had nearly 5 times the odds of developing asthma compared to those without exposure. The investigation involved participants from several European countries and noted that the negative respiratory effects were observed independently of other common allergens, such as those as house dust mites. In addition to the increased risk of asthma, roundworm exposure was also associated with lower overall lung function. As an entirely unrelated aside, what happened to the raw water trend? Tardigrades, commonly known as water bears or moss piglets, are microscopic animals that have become quite popular in recent years. Under stress, these organisms enter a dormant state known as the tun. In this state, tardigrades retract their legs, expel most of the water in their bodies, and drastically reduce their metabolism, thereby enhancing their survival under adverse conditions such as desiccation, freezing, and high radiation. They essentially turn themselves into a little piece of tardigrade jerky. I don't know about this one though. Homie may be dead. Tardigrades are technically considered polyextremophiles, meaning they can survive in many harsh conditions, but they are not classified as extremophiles because they don't thrive in them. Recent research has identified that the trigger for this transformation is linked to the oxidation of the amino acid cysteine. Experiments demonstrated that an increase in reactive oxygen species, particularly superoxide, accompanies the transition into the tun state. When oxidative stress is alleviated, tardigrades are able to rehydrate and resume normal activity. Urocentrum turbo, a ciliate belonging to a group of microorganisms that undergo chromatin elimination during cell division, was also observed in the samples. These unicellular organisms exhibit a large granular macronucleus and a compact micronucleus. They also sort of look like little balloons, and in my opinion are appropriately named. During the process of binary fission, which is a type of reproduction where cells split into 2 identical daughter cells, the macronucleus elongates and a distinct dark body forms at its center. As the cell divides, this body fragments into smaller portions that eventually disperse in the cytoplasm. Recent studies on related species, such as Glaucoma pyriformis, have shown that the discarded DNA is not merely wasted. Instead, it is repurposed to synthesize ribonucleic acid for their daughter cells. This mechanism ensures that each new cell inherits the necessary components for protein synthesis and genomic function. You going on the trail?